Welcome to clickmyproject.com. Let me discuss about the project unsupervised segmentation and classification of cervical cell images. Let me discuss about the abstract of the project. The proposed procedure ideas a tree using hierarchical clustering and then organizes the cells in a lined order by using an optimal leaf ordering algorithm that maximizes the parallel of adjacent leaves without any must of training examples or parameter regularization. Performance estimation using two datasets show the efficacy of the proposed tactic in images having uneven staining, poor contrast and overlapping cells. The cure rate is strictly related to the stage of the virus at diagnosis time, with a very high prospect of fatality if it is left untouched. The pap smear test is a manual screening procedure that is used to detect precancerous changes in cervical cells based on color and shape properties of their nuclei and cytoplasms. Automatic this procedure is still an open problem due to the complexities of cell structures. In this paper, we propose an unsupervised approach for the segmentation and classification of cervical cells. The segmentation process involves automatic thresholding to separate the cell regions from the background. A multi-scale hierarchical segmentation algorithm to partition these regions based on homogeneity and circularity, and a binary classifier to finalize the separation of nuclei from cytoplasm within the cell regions. Classification is posed as a grouping problem by ranking the cells based on their feature characteristics, modeling abnormality degrees. The proposed procedure constructs a tree using hierarchical clustering and then arranges the cells in a linear order by using an optimal leaf ordering algorithm that maximizes the similarity of adjacent leaves without any requirement for training examples or parameter adjustment. Performance evaluation using two datasets show the effectiveness of the proposed approach in images having inconsistent staining, poor contrast and overlapping cells. Let me discuss about the flow diagram for our process. First, to capture the input image from the dataset folder. Here we take the cell images and input, and in the background extraction, morphological operations such as erosion, dilation, opening and closing. Often combination of these operations are used to perform morphological image analysis. There are many useful operators defined in mathematical morphology. There are dilation, erosion, opening and closing. Morphological operations apply structuring elements to one input image, creating an output image of the same size. Irrespective of the size of the structuring element, the origin is located at its center. The watershed segmentation algorithm is an effective method that models the local contrast differences using the magnitude of image gradient, with the additional advantages of not requiring any prior information about the number of segmentation in the image. Automated and semi-automated screening procedure involve two main tasks: segmentation and classification. Segmentation mainly focuses on separation of cells from the background as well as the separation of nuclei from the cytoplasm within the cell region. Next, in the feature extraction stage, nucleus features are extracted. Nucleus area, the number of pixels in the nucleus region. Nucleus brightness, the average intensity of the pixels belonging to the nucleus region. Nucleus longest diameter, the diameter of the smallest circle circumscribing the nucleus region. We calculate it as the largest distance between the boundary pixels that from the maximum chord of the nucleus region. Shortest diameter, the diameter of the largest circle that is totally encircled by the nucleus region. It is approximated by the length of the maximum chord that is perpendicular to the maximum chord computed above. Nucleus elongation, the ratio of the shortest diameter to the longest diameter of the nucleus region. Nucleus roundness, the ratio of the nucleus area to the area of the circle corresponding to the nucleus longest diameter. Nucleus perimeter, the length of the perimeter of the nucleus region. Next, in the cytoplasm part separation. Cytoplasm features are cytoplasm area, the number of pixels in the cytoplasm part of a cell region divided by the number of cells in that cell region. We assume that the total cytoplasm is shared equally by the cells in a cell region. Cytoplasm brightness calculated similar to the nucleus brightness. However, overlapping cells are associated with the same value of the cytoplasm brightness. Cytoplasm maxima calculated similar to the nucleus maxima feature. Overlapping cells are associated with the same value. Cytoplasm minima calculated similar to the nucleus minimum features. Overlapping cells are associated with the same value. Cytoplasm or nucleus ratio. This feature measures how small the nucleus of cell is compared to the cytoplasm. It is given by the ratio of the nucleus area to the cell area, which is calculated as the sum of the nucleus and the cytoplasm area. Finally, we classify the both features and analyze the nucleus and cytoplasm part. After segmentation and classification mainly focus on automatic labeling of the cells into two classes. Support vector machines recently become one of the most popular classification methods. They have been used in wide variety of applications such as text classification, facial expression recognition, gene analysis, and many others. Support vector machines can be thought of as a method for constructing a special kind of rule called the linear classifier in a way that produces classifier with theoretical guarantees of good predictive performance. Machine learning is the process in which an algorithm improves or learns through experience. Machine learning has its roots in artificial intelligence and is a topic that has received increasing attention over the last few years. Finally, we got the analysis result. Let me discuss about the running procedure for our process. Select the main.m file in the current folder, right click that file and run for executing this process. First, initially the watershed segmentation process is performed. After that, feature has been extracted based on the nucleus and cytoplasm part. After that, based on the feature values, classifier is analyzed. The result of our cells. This is the original input pap smear image, that is cell image, and this is the background extraction of our result. This is the after extracting background result, we got the original image like this. And this is the watershed segmentation. After applying watershed segmentation, we got the result as like this. And this is the superimposed water segmentation. After segment the particular area, we have to superimpose the original image with that segmented result. This is the image labeled by cluster indexed, that is label the every region based on the cluster group, and this is the objects, that is a surrounding point in our cluster region. This is the cluster result, and that is second cluster group, and this is the third part of our cluster group. And finally we calculated the feature extraction values. This is the feature values for our input image, and finally we got the result as abnormal cell. Based on that feature values, we estimate the analysis by using the SVM classifier. Finally, both the segmentation and the classification perform better compared with existing system and provided the better analysis result of our input cell images. Thank you.